I'm Dr. Andrews, the founder and CEO of CellBridge Strategies, an international growth consulting firm that focuses on the life sciences and more specifically regenerative medicine. So I'm here today to introduce Dr. Jeanne Loring. Dr. Loring is a pioneer in human stem cell research. She is currently a professor emeritus at the Scripps Research Institute in La Jolla, California. She is also the founding director of the Center for Regenerative Medicine and serves on scientific advisory boards for academic institutions and corporations, including Merck KGaA. She serves on editorial boards for several journals, including Stem Cells, Translational Medicine, and Stem Cell and Development. Over the past 20 years, Jeanne and her group have had many firsts in human pluripotent stem cells, where they have reported on numerous transcription and translation regulatory factors in these cell types. Jeanne's group has most recently developed the PluriTest, a bioinformatic assay for pluripotency. Dr. Loring has also founded two companies, Apros Biosciences, which is now part of Biosite, and Aspen Neuroscience, which focuses on autologous neuron replacement therapy for Parkinson's disease. One of Jeanne's most recent projects is in collaboration with the San Diego Zoo, where iPS cells are used for pre-extinction of the northern white rhino. Which brings us to our discussion today, stem cell science and the pre-extinction of mammalian species. It's a great pleasure and honor to be here. And in our previous conversation, we spoke about what really started your career in human pluripotent stem cells and how that career has advanced over time into finding wonderful transcription factors that can be used not only in humans, but across species, how it's led into iPS cells, and most recently into how you use this technology for the pre-extinction of the northern white rhino. So if we can start at the very beginning, in 1999, let's go back.
Yeah, that's great. Thank you. It's not really the very beginning, but in 1999, having been in biotechnology for a number of years and also academics, I had a great interest in both genomics and pluripotent stem cells, starting with mouse. And then in 1999, I started a company that was making human embryonic stem cells from discarded embryos from IVF clinics. That was while I was still employed as I can't remember what my title was, senior scientist at, maybe director at Insight Genomics, where we were doing a lot of DNA sequencing. So what happened next was I had this very small company. We're essentially 4 of us altogether, including the person that worked for me. And we derived a fair number, that is 9 embryonic stem cell lines from human embryos. And we were not very well funded, but in 2001, George Bush announced that he would allow funding for human embryonic stem cells from the NIH by the federal government, and that changed everything. It changed things in some interesting ways, but the main thing it changed was that I could now apply for money from the federal government to stem cell research. And that was what launched me back into my academic career, which I've been in for the last 15 years, more than that actually by now, almost 20 years. So then I just started studying human pluripotent stem cells nonstop, and eventually started in 2007, started making human pluripotent stem cells out of fibroblasts that we had gotten from people's for biopsies. That was a reprogrammed technique that was developed by Shinya Yamanaka in Japan, and it won the Nobel Prize in 2012. It had a huge impact on the field, not only of human biology, but as I'm sure I will be explaining, also on the idea that you could use pluripotent stem cells to help rescue endangered species.
Yes, absolutely. That's a phenomenal scientific advancement. Just the thought of taking fibroblasts and differentiating them into an iPS cell and then redifferentiating them into the cell type of choice is just a phenomenal feat. And that technology is now, there's numerous clinical trials in progress in iPS cells. Can you tell me a little bit more about the significance of that finding and how you're using it moving forward?
Yeah, so induced pluripotent stem cells, iPS cells, are matched to the genomics of the person they're derived from. So that means that every single person on the planet could have pluripotent stem cells in a culture dish that are able to give rise to any cell type in the body. So a lot of those cell types are clinically relevant. In particular, the second company I started, Aspen Neuroscience, is using individuals' iPS cells to make dopamine neurons, which are the cells that die in Parkinson's disease. And the intention is to hopefully next year start implanting dopamine neurons derived from people's own cells back into their brains to restore function that is lost to Parkinson's disease. And along with that, there are other clinical trials underway, and there are clinical trials underway. There's one at the NIH that is using another derivative, retinal pigment cells from people's iPS cells to treat macular degeneration, which is an age-related loss of sight. So I think the sky is literally the limit here. There's a whole new world of medical applications that are just now starting to come into fruition.
So let's talk a little bit about transcription factors and how important they are in this science. Can you explain that a little bit?
Yeah, transcription factors are like master regulators, and I think we continue to be surprised. They bind to the working end of genes, the promoters, and they cause those genes to get turned on so that you're making a whole suite of different genes depending on what cell type you are. And it turns out that pluripotent stem cells are really governed by expression of a handful of transcription factors that set up their entire their physiology, their genetics, their pluripotency.
Is orchestrated, basically.
Is orchestrated, thank you. Orchestrated by those few transcription factors. And that's the reason why iPS cells are possible. And then moving forward from that, transcription factors turn genes on and off all through development. This is an embryonic development. They are the factors that elicit the expression of the right proteins that make a cell a particular type of cell.
It's very interesting. Very interesting. So I know you're working with the San Diego Zoo on the northern white rhino. And so why do you use the term pre-extinction? A lot of people have been using de-extinction for dinosaurs and all different other species. And why do you feel pre-extinction is a more valuable term?
Well, we want to, with a zoo, we want to catch animals before they're entirely extinct. And they are becoming extinct at a rapidly growing pace. The idea is that with pluripotent stem cell technology, you can take a cryopreserved sample of cells that were taken from an animal 20 years ago and kept frozen in liquid nitrogen all this time. And if you have cells, you have all the machinery in order to make stem cells, pluripotent stem cells. And then once you have pluripotent stem cells, you have all the potential to make any cell type. So the frozen zoo at the San Diego Safari Park, it's changed names a few times, but it's still the Institute for Conservation Research, has been harvesting and culturing skin fibroblasts from animals as they died, and some that are just captured in the wild and some that are in the zoo. And so they have thousands and thousands of samples of individual animals that are now cryopreserved as fibroblasts. So when I think it dawned on us one day that fibroblasts could be made into pluripotent stem cells, that was in 2007. And it dawned on us simultaneously in my lab and at the zoo. And so we thought fibroblasts, we should be able to make pluripotent stem cells. If we can make pluripotent stem cells from an animal before it's extinct, because it's still alive in the incubator, then we can essentially rescue it from the inevitable extinction because of loss of habitat and dwindling of the number of animals.
Right.
So we chose the northern white rhino, and we did this, I will confess, it was done simply because I asked Ollie Ryder, who's the head of the Conservation Research Institute there at the zoo, what his favorite animal was. And he said the northern white rhino. At the time, I think there were 7 left on the planet.
Wow.
And now there are only 2. They're both female. The animals are not quite extinct yet, but they can't reproduce. And so there is no way to rescue them. That's why we're calling it pre-extinction. We're rescuing them before they're entirely extinct. And part of that pre-extinction is the existence of these fibroblasts, these skin fibroblasts that have been frozen for a long periods of time from individual animals. So really what applies to people applies to animals. Those cells are they are alive. They continue to be alive even after an animal has died.
So that's why I was so interested in the transcription factors, because earlier you had said you were using some of the human transcription factors for the iPS cells in the rhino. And so it may be that you can go cross species with some of these technologies, which is fascinating.
It is fascinating. At the time, that was the only technique we had. I mean, we could have started all over, but the northern white rhino had not had its genome sequenced. So we couldn't find the right factors that were rhino specific. So we tried the human factors, transcription factors. And it took a little bit of fiddling, but not really. The transcription factors worked perfectly in the rhino. Delivering them was a challenge but it was a challenge for all cell types at that time. But once we got the transcription factors expressed in the cells, they turned into iPS cells. And we've done it with a lot of other animals too. And the first publication, which was the first publication on reprogramming, that is making pluripotent stem cells from any species. We also reprogrammed a highly endangered primate called the drill, using the same approach, exactly the same approach. So what that means is that this probably can be done with we know it can be done, maybe with some fiddling, for virtually every mammal. There may be exceptions, but it probably can also be done for other animals that are not mammals. It's not clear how hard that's going to be once you start expanding beyond the mammalian kingdom. But it really is. I think what it is it's a spark that has generated a lot of people thinking about how they might be able to rescue a frog or a snake or something else that's died. And it's really the same principle. As soon as you realize that there's another way that you might keep this animal from becoming extinct, then you start expanding and looking at other scientific approaches that you never thought of before.
So you've been able to take the iPS cells and redifferentiate them into oocytes. Is that correct?
We haven't made oocytes yet. Yes, we have. Let me put it this way. We've made the precursors for oocytes, which are called primordial germ cells. Yeah. But we can't make them exclusively. We can make them along with a lot of other cell types. So we know they're capable of making them. But what's being worked on now at the zoo, under the leadership of a woman who trained in my lab, Marisa Karodi, she is now working on developing methods to make real mature oocytes so they can be fertilized by sperm. And it really is just like the methods that we use to make any cell type, like the neurons we're making for Parkinson's disease. It's a tedious process. It's trial and error, but we have an advantage now. Just this year, we published the entire annotated genome sequence of the northern white rhino. And once you have the sequence, then a lot of doors become open for example, identifying the right transcription factors that might push the cells in becoming oocytes. Without that tool, it's very, very difficult. And sometimes we forget, because the human genome, of course, has been the focus of a lot of interest and has been sequenced thousands and thousands and thousands of times. I mean, my own genome has been sequenced 3 times. So, you know, why not?
Why not, right?
The technology got better, so I said, let's do it again. And it's amazingly useful to know the whole map of the genome. Then you can start using genomic techniques to modify cells so that they can become different cell types, and so it really opens even more doors. And so that's available as of this year.
Fantastic. Has anyone taken iPS cells back to oocytes for any?
Yes.
What species?
For mice, for laboratory mice. So we know once we find that something's possible in mice, we can often, but not always, figure out a way to make it possible for humans, which is always the next step people take. And if it's possible for humans, or at least looks promising for humans, then you can imagine expanding to all sorts of other animals. There is a company in the Bay Area that is actually trying to do that with human iPS cells to make oocytes, and so it's almost a parallel process. I'm interested in what they learn, and I want to apply it to the rhino. They're interested in what we learn from the rhino. They can apply it to humans.
Absolutely, absolutely. And then you can apply it to other animals along the evolutionary chain. So the rhino is very closely related to the horse.
Yes, yes. And it's certainly, I know this is going to be feasible in the horse.
Yes.
There are some animals that have been recalcitrant to reprogramming, but what we've learned is that you just need to keep modifying the process over and over again, and eventually, and hopefully not too long, you'll figure out how to do it, because we know it's possible.
Very interesting. Very interesting. So if you were to think about your wonderful career and all the things that you've achieved, what would you like to see happen in your lifetime that comes out of your work?
Well, obviously, I want to see a cure for Parkinson's disease and hopefully other diseases that I've studied. I mean, when I say a cure, I mean a therapy that reverses the symptoms of the disease. It doesn't remove the disease. You can't remove diseases. But the other thing is, I'd love to see a northern white rhino, pure white northern white rhino calf born. The zoo is set up to do this. The zoo has brought in, and it was an interesting story in itself, how you bring in on an airplane 6 northern white rhino females from Africa, and you deliver them to the San Diego Wild Animal Park. And then those animals have a special enclosure, and they're being studied relentlessly to understand their reproductive system, so they can, these are southern white rhinos now, closely related to, but not exactly like northern white rhinos. Those animals are meant to be the surrogate mothers for the northern white rhino embryos that we can generate in the lab. And by the way, generating those cells, those embryos in the lab is the same process that's being used to make human embryos in vitro fertilization clinics, assisted reproduction. All those methods that have been developed for humans can be applied to the rhino.
So we spoke earlier about the need for spermatozoa.
Mm-hmm.
And theoretically, you could create you could even use ICSI and create a sperm cell and use intracytoplasmic sperm injection to create an embryo that way.
Yes. And making sperm cells shouldn't be any harder than making oocytes. I mean, it probably is, but really all it has to do with is, and with sperm, you don't really have to have them, you need to have them be haploid, because each one of the sperm and the egg undergo a special cell division called meiosis. And if they need to do that in order to get rid of half their chromosomes, so that when they combine again together in an embryo, you still only have the diploid number of chromosomes. So if you can make a cell that has the qualities of a sperm, in that it's haploid and it has its DNA wound up right and everything, you should be able to inject the nucleus right into the oocyte. But there are stored sperm from northern white rhinos. That was pretty easy. You could get mature cells. And so the first step will be to make oocytes out of the iPS cells and then fertilize them with the stored northern white rhino sperm.
Fantastic.
Yeah.
Fantastic. Well, such phenomenal technology. Very interesting. Jeanne, is there anything else you'd like to talk about with regards to your companies or any of your current wonderful announcements that have come about lately?
Yeah, there actually was a really good, my company, Aspen Neuroscience, just raised $147 million
Congratulations.
to take the Parkinson's disease therapy through at least the phase 1 clinical trials. So I'm hoping that that's imminent. It's in the hands now of the medical people and the FDA, but I don't see, you never know, I don't see any real blocks to that proceeding, at a reasonable rate. And I'm hoping that the first patients get treated next year. Early next year would be preferable for me. That, and I'm also involved in the space program at NASA on the International Space Station. I'm interested in knowing what happens to astronauts' brains in space. So
that's a very good question.
Yeah, because we don't know. I mean, we have lots of evidence that maybe something goes wrong, but it's really hard to measure in space. So we're sending my colleagues and I from the New York Stem Cell Foundation and from the National Stem Cell Foundation are making these structures called organoids, which are made up of simple parts of the brain. One kind of neuron or just a few types of neurons, and then adding the immune system of the brain, which is called microglia, and wrapping them all up in these tiny little spheres that are about 0.2 of a millimeter across. So they're really tiny, but they have components of the brain. We've been sending those up into space to the International Space Station. We plan to do that one more time and many more times, but the next time will be on June 7th this year from Cape Canaveral, and I'm planning to be there to watch the rocket launch.
Fantastic. Fantastic. So are they in a bioreactor, the organoids?
Well, they're actually only keeping warm in a static culture environment, which we developed especially for this. They're going to be on the space station for 30 days. So that's, and we've shown that they are perfectly healthy in 30 days in a static culture system. So we're doing that again this time.
Fantastic.
We're hoping to get more sophisticated in the future so that we can change the conditions and monitor them better in space. But for now, we know this will work and it will give us information about the effects of microgravity. And obviously, if there is increased radiation, that will also be in the mix. So far on our last flight, which was in 2020, 20, there we go, way back. We discovered that we found out that there wasn't a lot of excess radiation in the vicinity of the incubator they're in, which is just keeping them at body temperature. So we don't think there'll be a big cause of a big change because of radiation, but we have to monitor that along with temperature. They have to go up in a rocket and then come back down and splash, and they have to travel on trucks in order to get to the place where they get analyzed. So we have to take all of that into consideration. And we do that by having a set of organoids, a set of samples on the ground that stay on the ground.
Yeah.
And so everything is done not as an absolute, but as a comparison to what would happen to them if they had been on the ground and not flown into space.
That's fantastic. You've got direct ground truth data.
Yeah, that's right.
For each experiment, which is wonderful. And
all this, I'm just going to say all of this is because I love stem cells. And so I can't resist using them in another way. Yeah.
It's fantastic. It's that you're creative and you're using the knowledge you have to go in many directions because you're inquisitive and you love the science. And that's wonderful. Well,
thank you very much, Shane.
No, sure. Thank you very much for your time today. And I've really enjoyed speaking with you. And if you have any questions, let us know in the future and we'll be happy to be there for you.
Okay. I really appreciate it. Thank you so much. It was a pleasure to talk with you. And I want to thank Bernie Siegel for making this possible.
Yes. I thank Bernie Siegel as well. Thank you. Okay.
All right.
Goodbye.
Goodbye.